Ava is a rising junior at St. Andrew's School in Boca Raton and this summer intern in the bio laboratory. Ava has a passion for biology, chemistry, and computer science, and as others in this group has demonstrated this by taking advanced courses in these subjects, and Ava's goal is to pursue graduate school after completing college.
Hello, everyone. My name is Ava Allwardt, and I had the privilege of working in the bio laboratory this summer. My project focuses on elucidating the structural dynamics of protein sec using technique HDX-MS. The bio laboratory develops nanoparticle technology to probe the interactions of proteins on the cell membrane. Membrane proteins are proteins that perform a variety of cell functions that are located on the phospholipid bilayer of cells. The bio laboratory looks at membrane proteins as potential antimicrobial targets. This is especially important because antibiotic resistance is on the rise, and we need to find alternatives to antibiotics. Membrane proteins provide a viable alternative. However, membrane proteins are often challenging to study. This is because they do not perform the function that they do in their native environment outside of the cell membrane. For this reason, the bio laboratory uses the nanoparticle tools it develops to study how membrane proteins undergo conformational changes, the regions that participate in those conformational changes, and how we can target those regions to disable the protein function and result in bacterial cell death. The specific protein that I used in this study is called sec-A. Sec-A is a peripheral membrane protein that translocates unfolded peptides across the bacterial cell membrane. Sec-A works as an ATPase, meaning it breaks down ATP into ADP to provide energy for this reaction. Sec-A interacts with the sec-YEG complex and sec-B, which are other two proteins, as well as lipids to allow for this translocation. Sec-A is essential for cell survival, meaning that if it cannot perform its function, the bacterial cell cannot survive. For this reason, we want to understand the conformational changes that sec-A is undergoing, specifically when it is bound to lipids and interacting with those lipids, so that we can target those regions, disable them, and result in bacterial cell death. The tool that we are using to understand sec-A's conformational changes and structure when in its unbound state compared to its bound state is called nanodiscs. Nanodiscs are artificial discoidal lipid bilayers, and they have two components. These include phospholipids, which include the phosphate head and the fatty acid tail, and membrane scaffolding proteins, which encircle these phospholipids. Nanodiscs provide a medium upon which sec-A can interact with lipids in an environment outside of its native environment and undergo the same conformational changes that it would undergo if it were bound to a real cell membrane. This allows us to study the conformational changes that sec-A undergoes, understand the regions that are interacting with lipids when it is bound to the nanodisc, and then be able to target those lipids to use sec-A as an effective antimicrobial target. The technique that we are using to understand the conformational changes sec-A undergoes when bound to lipids is called HDX-MS. HDX-MS stands for hydrogen deuterium exchange mass spectrometry. Deuterium is an isotope of hydrogen, meaning it has a different number of neutrons. Specifically, deuterium has one additional neutron. This means that a peptide that is covered in hydrogen will have a slightly lower mass than the same peptide covered in deuterium. This allows us, or HDX-MS uses solvent accessibility to allow us to understand which parts of sec-A are able to uptake deuterium or which parts are exposed when soaked in a deuterium-based solution. Parts of the protein that are exposed to the solution will uptake deuterium at a faster rate, while parts of the protein that are protected or enclosed inside of the protein will not be able to uptake deuterium at the same rate. When a protein undergoes conformational changes, the parts of the protein that will be exposed to the solvent will change, and this is how we are able to compare deuterium uptake and thus the structure of sec-A when it is bound to nanodiscs compared to when it is not. The steps of HDX-MS are as follows. The first step is to take our protein and soak it in a deuterium-based solution. This allows the hydrogen atoms that are on parts of the protein that are exposed to the solvent to be replaced with deuterium atoms. As you can see in the image, hydrogen atoms on parts of the protein that are protected by other parts of the protein are not able to be replaced with deuterium. We record this at different times in order to evaluate the extent of deuterium uptake for each peptide in our protein. The next step is to quench this reaction. To do this, we use a buffer with a low pH and a low temperature. This allows us to stop the reaction as well as prevent a back exchange where deuterium atoms are again replaced with hydrogen atoms, which would then skew our data. The next step is the proteolysis step. This is where we use a protease, which is an enzyme that cleaves proteins, to cleave sake into individual peptides. This allows us to then run those peptides through mass spectrometry. When a peptide is up to deuterium and its mass is increased, its envelope on the mass spectrometry graph will shift, indicating that it has a greater mass than before, or when it was not soaked in deuterium. We analyze the mass spectrometry graphs for each peptide, and then we are able to generate an uptake graph to map deuterium uptake for each peptide over all time points. The aims of this research project were to identify the regions of sake that participate in interactions with the lipid bilayer. We predict that the N-terminus likely plays a role in sake-lipid interactions due to previous research. However, we are also looking for other domains of sake that may initiate or participate in these interactions with lipids. This will allow us to harness sake's potential as an antimicrobial target, because if we can understand the regions of sake that participate in these interactions with lipids, we know which regions we need to develop a molecule to essentially disable those regions from binding to lipids and thus disable sake's function and cause bacterial cell death. We also hope to develop the novel nanodisc-based approach. Currently, in the early stages of the study, we are using smaller nanodiscs, which are around 10 nanometers in size. However, once establishing this protocol, we plan to scale up to larger nanodiscs, which are about 20 nanometers in size. Larger nanodiscs have not been used with membrane proteins, and so this will be a novel approach to understand sake's conformational changes with lipids. The first preliminary experiment that we performed was a digest test with protein sake. This was used to determine the ideal protease and quench buffer combination for use in HDX-MS. We tested 7 buffers and 2 proteases, including pepsin and NEP2. We did this by running the protein, the buffer, and the protease through mass spectrometry, and then analyzing the results using mascot data analysis. We found that 0.1 molar of Na-phos-TSAP, when combined with pepsin protease, provided the maximum sequence coverage of protein sake at all cutoff values. 20% cutoff is only shown in this image. However, 0.1 molar Na-phos-TSAP when combined with protease did provide maximum sequence coverage from all cutoff values tested, ranging from 0% cutoff to 20% cutoff. Thus, we could determine that this was the right protease and buffer combination for use in HDX-MS. The next step was to run HDX-MS with 2 samples. This is sake in its unbound state and sake when it is bound to nanodiscs, so that we can compare the interactions that sake is undergoing and the conformational changes it is undergoing when it is bound to nanodiscs to when it is not. To do this, we cultured E. coli cells and then purified the sake protein from these cells. We took part of our sample and reconstituted it with nanodiscs. This gave us our 2 samples, sake in its unbound state and sake when bound to nanodiscs, to run through HDX-MS. We ran these 2 samples through HDX-MS and mass spectrometry graphs for each peptide, for each time point, and for each bound and unbound state of sake were generated. We analyzed mass spectrometry graphs for each peptide in both states of sake. One of the main things that we look for when analyzing mass spectrometry graphs is to ensure that the envelope of each graph is symmetrical. This ensures that our data is accurate and can be used for further analysis. We also look at our deuterium uptake graph. We look to make sure that percent deuterium uptake at T0 or time point 0 is near or at 0. This is because if deuterium uptake was higher, this would not quite make sense because we have not soaked the protein in deuterium yet. We also look to make sure that the uptake graph has a positive or a neutral slope, because a negative slope would indicate back exchange where deuterium atoms were being replaced again with hydrogen atoms, and that would also skew our data. After analyzing the mass spectrometry results using HDX workbench for each peptide, for each time point, and for each state of HDX-MS, we are generated with 2 sequence coverage maps. The sequence coverage map on the left represents the deuterium uptake for each residue or amino acid in sake in its unbound state. The sequence coverage on the right represents the same for sake when bound to nanodiscs. Cooler colors indicate regions of sake that had a lower deuterium uptake, while warmer colors indicate regions with a higher deuterium uptake. As you can see, these sequence coverage maps look relatively similar. However, there are subtle conformational changes that we notice. For example, here at around residue 710 to 720, which corresponds to the helical wing domain or HWD on sake, we see that there is more deuterium uptake when sake is bound to nanodiscs. This indicates that there is likely a conformational change occurring when sake is bound to nanodiscs that exposes this region more to the deuterated solvent. Here's a comparison map showing the deuterium uptake of sake versus sake when bound to nanodisc in one sequence coverage map. Gray regions indicate amino acids where deuterium uptake was the same for sake in its unbound state and sake when bound to nanodiscs, while colored regions indicate places where the ligand when bound to nanodiscs differed from sake in its unbound state in regards to deuterium uptake. Then we were able to map this onto a 3D structure of sake using Chimera. The same region that we noticed before that had higher deuterium uptake on sake when bound to nanodiscs corresponds to this helical wing domain on this region of sake. This indicates that when sake is bound to nanodiscs, this region is opening up, likely to allow ATP to enter and allow for the ATP hydrolysis reaction to take place. When this region opens up for ATP, these residues and this peptide becomes more exposed to deuterium, and thus we see this result of when bound to nanodiscs, sake has more deuterium in this region. In conclusion, we found that the helical wing domain, or HWD, likely plays a role in sake-lipid interactions. Interestingly, we identified a region other than the N-terminus, and we will use further studies to confirm these findings. Primarily, we will bind sake to larger nanodiscs, and when we do this, we expect that sake will undergo more dramatic conformational changes. As I said, we used around 10 nanometer nanodiscs in this experiment, and we found that although there were subtle conformational changes, we want to see larger conformational changes to cement our findings. We also plan to collect data from various structural determination methods, which means in addition to HDX-MS, we plan to use cryo-EM as well as negative stain EM to fully understand which regions of sake are undergoing conformational changes when bound to lipids, such as the HWD or the N-terminus, and really understand how sake can be an effective antimicrobial target. Once we understand the regions of sake that are participating in these interactions, another lab will develop a drug to inhibit these regions of sake, inhibit it from binding to lipids and performing its necessary function for the cell, and thus the bacterial cell will result in cell death. I would like to thank the Mao Laboratory, especially my PI, Dr. Juan Mao, for accepting me into his lab and for all of his support. I'd also like to thank my mentor, Sarah Dallow, for all of her guidance and support throughout this internship. I would like to thank the Griffin Laboratory for guiding my mentor and I through the use of HDX-MS, especially Dr. Tim O'Leary, Scott Novick, and Valentine Karubel. I would also like to thank our program coordinator, Dr. Rosie Albrand-Zeckler. And of course, thank you to the William R. Kenan Jr. Charitable Trust for making this experience possible. Does anyone have any questions? Yes?
Because the nanos is so small, what does it physically look like when you're working in the lab?
So we actually cannot see it. The only way that we could see it would be through cryo-EM or through negative stain EM or another imaging technique. But essentially we're dealing with small tubes of clear liquids, but we do know that sake is binding to nanodiscs because when we run it on an SDS page gel, we see the band appear where the mass of sake when bound to nanodiscs should appear. Thank you.